Hey everyone, and welcome straight into today's explainer. Today, we're taking a whiteboard-style approach to literally one of the most fundamental processes on planet Earth, photosynthesis. So I'd highly recommend grabbing a pen and some paper, because we are going to sketch out the absolute essentials you need to know. We're talking the core science, the required practicals, and exactly how to crush this topic inside and out. All right, let's just dive right in. So here's our game plan. We're going to start with the core equations, peek inside a plant leaf, and figure out what actually slows this process down. Then we'll look at the required practicals, see how plants use their newly made glucose, and finish up with some classic exam traps to make sure you get those top marks. All right, part one, the core equations, the recipe for plant life. At its most basic, everyday level, photosynthesis is just how plants make their own food using energy from light. The word equation is pretty straightforward, right? Carbon dioxide plus water, add some light, and you get glucose and oxygen. But the real magic, the thing you absolutely need to memorize, is the balanced symbol equation. It's 6CO2 plus 6H2O, which reacts to form C6H12O6 plus 6O2. When you're jotting this down from memory, those balancing sixes are absolutely crucial. It's the exact molecular recipe that makes plant life possible. Now, let's zoom in a bit and see where this actually happens. Imagine drawing a microscopic plant cell. Deep inside that cell, you've got these structures called chloroplasts. A chloroplast is the specific organelle, literally the exact microscopic location in the cell where all this chemical magic takes place. Think of it as the plant's tiny food factory. But inside that chloroplast, we have something slightly different with a very similar name. If we were coloring this in, we'd shade it bright green. That green stuff is the pigment called chlorophyll, and its one job is to absorb the light energy we need to kickstart the reaction. So here's the crucial difference. The chloroplast is the place where it happens, and chlorophyll is the pigment that traps the light. Trust me, mixing these two up is a classic mistake, so definitely keep them separated in your head. Moving right along to part two, inside the plant leaf, the perfect microfactory. If we transition from a single cell to looking at a cross-section of a whole leaf, it's wild how perfectly adapted it is. First off, a leaf has a huge surface area just to catch as much sunlight as possible, and it's super thin, which allows gases to diffuse really quickly. At the very top, you've got this transparent layer called the upper epidermis. It's literally like a skylight, letting the sun shine straight through to the palisade layer underneath, which is just absolutely packed with those chloroplasts we talked about. Further down, the spongy mesophyll has all these air spaces to let gases move around easily. And finally, on the underside, we have stoma, tiny little pores controlled by guard cells that act like doorways. They let the carbon dioxide in and let the oxygen out. Okay, part three, understanding the limiting factors. What slows down the process? We know the recipe, and we've seen the factory. But out in the real world, environmental conditions aren't always perfect, right? So what happens when something acts like a brake pedal? We call that a limiting factor. The textbook definition, which is a huge high-yield keyword for you, is something that slows photosynthesis when it's in short supply. Basically, if a plant has all the light and water in the world, but barely any carbon dioxide, well, that lack of carbon dioxide becomes the limiting factor. If you were to sketch out quick line graphs for these, light intensity and carbon dioxide would both look pretty similar, a steady climb that eventually flattens out. Less light or less CO2 just means a slower rate. But temperature is completely different. Because photosynthesis relies on enzymes, right? At low temperatures, enzymes are sluggish. As it warms up, they hit their optimum temperature and work incredibly fast. But, and this is absolutely vital, if it gets too hot, the graph literally drops off a cliff. The enzymes denature, their active sites get warped out of shape, and the whole food-making process just totally shuts down. Which brings us to part four, required practicals and proof, testing the theory in the lab. So, it's one thing to talk about formulas and theory, but how do we actually scientifically prove that photosynthesis just happened in front of us? You can't exactly watch glucose molecules popping into existence. But here's the trick. We know that when a plant makes a bunch of glucose, it stores the leftovers as starch. So, logically, if we can prove there's starch inside a leaf, we have concrete proof that photosynthesis occurred. And here is the exact step-by-step lab setup you need to know. First, we take the leaf and boil it in water. This kills it and stops any ongoing chemical reactions. Second, we heat it in ethanol. This strips away the green chlorophyll so we can actually see the color of our chemical test later on. Now, really important safety note here. Ethanol is highly flammable. You literally never put it near an open flame. You always use a hot water bath. Third step, we give the leaf a quick rinse in warm water just to soften it back up. And fourth, the main event. We add a few drops of iodine solution, which is our trusty test for starch. When that iodine hits the leaf, you're looking for one very specific color change. If the iodine turns a really dark, deep blue-black color, bingo, starch is present. That's a positive test, meaning photosynthesis definitely happened. But if it just stays its normal orange-brown color, there's no starch. You really want to cement this rule in your brain. Blue-black equals starch. We can actually use this same test to prove exactly what is needed for the reaction. If you tape some black paper over part of a leaf to block the sun, and then do the iodine test, that covered part stays orange-brown, the uncovered part turns blue-black. Boom, you've just proved light is required. Or take a variegated leaf, the ones with white patches that don't have chlorophyll and green patches that do. Only the green areas will turn blue-black, and that brilliantly proves that chlorophyll is absolutely essential for the whole thing to work. Now, what if we want to measure the actual speed or the rate of photosynthesis? Enter the classic pondweed experiment. You put some pondweed in a beaker of water and shine a lamp right at it. Because the plant is entirely underwater, the oxygen it produces escapes as little visible bubbles. By literally just counting how many oxygen bubbles pop up per minute, or by measuring the volume of gas in a syringe, you get a clear mathematical measurement of the rate. If you move that lamp closer to increase the light intensity, you're going to see a lot more bubbles. Moving on to part 5, how plants use glucose beyond just food. So the plant worked hard to make all this glucose. Now what? Where does it go? Picture a flowchart starting with one glucose molecule, branching out into 5 different jobs. Number 1, it's used right away in cellular respiration to release energy. Number 2, as we just saw, it's stored away as insoluble starch for a rainy day. Number 3, it's converted into tough cellulose to build strong cell walls. Number 4, the plant combines the glucose with nitrates it pulled from the soil to make amino acids, which then build proteins. And number 5, it's stored as fats and oils, which you'll often find in seeds. Examiners absolutely love testing these 5 pathways, so make sure you've got them down. And finally, part 6, exam traps and key points, maximizing your marks. Let's treat this like a one-on-one tutoring session for a second. When you sit down and face a heavy 6-mark question asking you to describe photosynthesis, I want you to mentally check off these exact points. You state it happens in the chloroplast. You state chlorophyll absorbs the light. You explicitly name carbon dioxide and water as the reactants and glucose and oxygen as the products. You absolutely must mention the energy transfer, that light energy is converted into chemical energy. And finally, wrap it up by saying the rate is controlled by limiting factors. Hitting those precise scientific beats is exactly how you lock in a perfect 6 out of 6. Now look at this phrase right here. Plants get their food from the soil. If I had a giant red marker right now, I would completely cross this out. It is the ultimate exam trap. Plants absolutely do not get their food from the soil. They make their own food, glucose, from scratch. The only things they absorb from the soil are water and mineral ions, like nitrates. Never ever write that soil provides food for a plant. All right, let's do a really quick rapid-fire recap of the whole explainer to lock this in. It happens in the chloroplasts, where chlorophyll grabs the light. It takes CO2 and water and turns them into glucose and oxygen. That glucose gets stored as starch. We test for that starch using iodine, and if it's there, it turns blue-black. In the lab, we safely use a hot water bath and ethanol to strip out the chlorophyll. And lastly, remember your limiting factors. Light, CO2, and temperature act as the brake pedals for the whole process. We have covered a massive amount of ground today, from the microscopic anatomy of a leaf right down to the safety steps of the iodine test. But before we go, I want to leave you with a challenge. You know the practicals, you get the limiting factors, and you know exactly what glucose does. But can you perfectly write out and balance the symbol equation for photosynthesis from memory right now? Grab your pen, write it down, and prove to yourself that you've mastered this. Thanks so much for hanging out and learning with me today, and I'll see you in the next explainer.